Hello friends, Ace Botane next episode. In this episode, we'll be discussing NCERT quick reference. Chapter is Molecular Base of Inheritance, third part. And what all things we'll be discussing in this episode, that is central dogma, reverse central dogma, packaging of DNA, both in prokaryote and eukaryote. Meanwhile, detailed note I already published in YouTube. This is for quick reference. Similarly, 2 questions daily I am putting. So to get all these, subscribe Ace Botane. Like it, share it, comment on it. So we can continue for convenience to save time to more efficiency, to reach more efficient manner. I will share the screen, so it'll be easy for you. We can save time also. I think screen came, so we can proceed. Remember, all references based on NCERT Reprinted edition March 2024. So we can proceed. Topic I already told you, so we can, without losing time, we can proceed. So central dogma and reverse central dogma. So what is meant by dogma? Dogma means an established suggestion or opinion. That is the meaning of the term dogma. Now, dogma means an established suggestion or opinion. Now here in genetics, molecular basis, Francis Crick proposed central dogma in molecular biology. Francis Crick. Now some photographs are given there, so it is clear to you, I think, who received Nobel Prize, all these items. Francis Crick received in 1962 in connection with the structure of DNA along with the Watson. Now this one, central dogma, again connected with the Francis Crick. This is Francis Crick, 1958. Idea is, that is DNA. Here you can find replication symbol is given. It's a famous chart given in NCERT. Then transcription, mRNA will be produced. Here you can find from mRNA, proteins will be formed. So there will be unidirectional flow of information. See here. And enzyme involved is transcriptase, important one. And this diagram given in NCERT. Page number 83, March 2024, reprinted edition. Now reverse of it. That is, in some viruses, the flow of information is in reverse direction. Viruses only. That is from RNA to DNA. And NCERT asks a question to us, can you suggest a simple name to this process? And that is what is known as reverse central dogma. That is why given an arrow there. Connected with the 2 famous scientists, Temin and Baltimore. Connected with the virus, Rao sarcoma virus, RSV, having single-stranded RNA. Now photograph you can find there, 1970. Both they proposed it, 1975, again they received Nobel Prize. Here, first is, chart is DNA, RNA, protein. Starting with RNA to DNA, reverse transcription. Then, second, DNA to RNA transcription. Anyway, later, translation and protein will be synthesized. So here you can find 2 steps. Here only one. That is central dogma and reverse central dogma. I think I did clear. Both are a neat point of view, important questions. Now, packaging of DNA helix. See, automatically NCERT asks a question to us, why there is a need to pack DNA? Size of nucleus is approximately 5 to 25 micrometer. NCERT value approximately is 10 raised to minus 6 meter. That is page number 83. Prokaryotes, in E. coli, the length of the DNA is 4.6 into 10 raised to 6 base pair. And when we calculate it, approximately comes to 1.36 millimeter to 1.6 millimeter. Now, packed in a cell of size approximately 1 μm. So it has to be packed in such a small sized area. Now, eukaryote, you can find mammalian cell, for example, 6.6 into 10 raised to 9 base pair into 0.34 into 10 raised to minus 9 meter per base pair. And that comes around almost 2.2 meter. And nucleus is approximately 10 raised to minus 6 meters. So NCERT value, popular question. Thus, long DNA has to accumulate in a small area by packaging in a suitable way. That is by compaction. Togetherness, compaction. Now, DNA packaging in prokaryote, detailed manner. Here, no defined nucleus in prokaryote. DNA is not scattered throughout the cell. So, no defined nucleus at the same time, DNA is not scattered throughout the cell. Seen in cytoplasm. DNA is in supercoiled structure and DNA is negatively charged due to the phosphate groups in the nucleotides. The maintenance of these coils are done by non-histone basic chemicals, non-histone. And that include polyamines with the positive charge. So charge can be attracting each other. The total structure packed, that is DNA, is called genophore or nucleoid. These are very important. DNA in nucleoid is organized in, next term NCERT important one, large lobes. Here they will give small loops. Large loops held by proteins. Now, DNA packaging in eukaryote. Here, complexity is more. Reason there will be developed nucleus at the same time, cytoplasm will be different. Arrangement is carried out by positively charged proteins called histones. Positively charged proteins called histones. A protein acquires charge depending upon the abundance of amino acid residue with the charge site chain NCERT page number 83. So, very important for NEET exam, statement-wise NCERT. That's why given there with the page number. Now we can continue. So, what is histone or what are histones? Histones are water-soluble basic protein. Histones are rich in basic amino acids. Another important question: arginine and lysine with the charged side chains. With 5 types of histone: H1, H2A, H2B, H3, H4. Out of these 5 types, 4 of them occurs in pairs. So total 8 forms histone octamer, otherwise called new body. The positively charged histone octamer is surrounded by DNA. It's negatively charged. This is positively charged, and they form what is known as nucleosome. And this diagram, again, a famous diagram from NCERT, labeling question, or core of histone molecule, H1 histone, histone octamer DNA. Then beads are nucleosome, and that it is a little bit expanded here. So NCERT diagram, one I drawn there for your convenience. We can proceed to the next page. This is originally an old NCERT data, the diagram, but in NCERT thing it is not there. So there they given the size, everything. Now, a simple question they given in NCERT, that is, when we make an approximate calculation, 1 bead equals 200 base pair. There is a calculation for it. For timing, 1 bead is 200 base pair. So 6.6 into 10 raised to 9 base pair into 1 by 200. That means it will be around 3.3 into 10 raised to 7 beads. So in mammalian cell, approximately 3.3 into 10 raised to 7 beads will be this is NCERT question, page number 83. Now, packaging of chromatin in higher level requires additional set of proteins called as non-histone chromatin, NSA protein, and different types are seen. 2 types of chromatin available. 2 types of chromatin available. So previously here, non-histone chromatin. Here, what we are discussing, 2 types of chromatins available. There's another usual question for convenience and for save time. I put a table there so we can save time also. 2 types have been identified or separated on the basis of staining nature and a typical nucleus chromatin. 2 types chromatin. Euchromatin, heterochromatin is a famous question. Very famous question. Repeated question. This year also we can anticipate. I think I get clear. With a minimum time, I tried to cover entire portion. Okay. And if you like this episode, like it, share it, comment on it. We can meet later. We'll be continuing the series. For time being, we can wind up. Take care. Bye. Thank you.